Okay. Good evening, Satish and SD. We are here for our introduction to proteomics course. I hope you all can see my slides as well. Yes, is it visible? You can give me a thumbs up like yes, it is visible and I can go ahead. Yeah. Okay. So good evening. Yeah, this is the NPTEL course. It has a code of BT60, which is Introduction to Proteomics. So we are here in our week 4 lectures. So in all the 3 weeks, we have went through a bit of gel-based proteomics. We have given introduction to what proteomics is all about, and like giving an introduction of proteins, amino acids, etc. How exactly we need to prepare samples for doing proteomic analysis. Then we studied one of the gel-based proteomics technique, like that is 2D gel electrophoresis. Now we'll be studying another gel-based proteomics technique, which is differential gel electrophoresis in this week. And along with that, we'll be studying a bit of system biology as well. So in this specific week 4 lectures, we'll be dealing with, so there were 5 videos which were uploaded. If you have all gone through, it is well and good. If not, I'll be just going through all those 5 lectures like in a quick way, and then we can go for a question and answer session. Like in an hour, I'll try to complete all of these 5 parts of the lecture. So wherein in the initial we'll be talking about just what dice technique is all about, how do we perform that. Then after performing that specific dice analysis, how dice technique, how can we analyze the data after obtaining the results, and a few applications related to that, how dice can be utilized. It is similar to that of 2D, but depending upon your need, you can use dice technique as well. And a bit of system biology and which is related to proteomics. The system biology is overall, like if you are studying the whole biology of a system, may it be human system, may it be bacterial system, may it be fungal system, whatever. Any system you are studying each and every perspective of that in an integrated manner. That's system biology. Like if I just give you an overview of what system biology is, that's what system biology is all about. So how exactly those type of study is done, etc. We'll be dealing with that in part 4 and part 5 of this lecture. So starting with Deitch. So what Deitch is all about? So this was the technique first reported by Unlu in 1997, and it is a technique wherein um, gel to gel variability is reduced basically. So if you see in terms of 2D gel electrophoresis, which we did in our last week lecture, so in that you can run one sample, like do the isoelectric focusing, and then do SDS page and then visualize. So you can run one sample, and per sample you will need a specific gel altogether. Whereas in terms of dice technique, you can use multiple samples. Like so it is basically a multiplexing technology, wherein 3 samples with the help of 3 different dice, they can be stained with those 3 different dice. We can pool them all together and then load it onto the sample. Now we can uh, so all 3 samples have been pooled together, and instead of using 3 different gels, we can use only one gel itself and do the analysis by visualizing those 3 samples differently because of some excitation and emission wavelength, because of the dyes which we have involved, like included in the technique. So the only difference between 2DE and DIGES in terms of workflow, you initially label the sample and pool it, whereas in terms of 2DE, you directly load your sample and do that 2DE analysis. Whereas in terms of DIGE, the first step involves labeling, and then you do a similar protocol like 2DE itself. So this is a protein labeling and a separation technique. So this is something which is added in DIGE technique, and it detects on the basis of fluorescence. So we have whatever labeling is done with fluorescently tagged labeled, Some of the commonly used is this side dice, side 2, side 3, and side 5, which has an hydroxysuccinylimidyl ester. It's a derivative of cyanide dyes itself, and these dyes are used in terms of this specific technique, wherein size and charge match. So basically, this all three dyes are having similar molecular weight, and they can co-migrate within the gel itself. So because of their similar molecular weight, it is not affecting the ultimate molecular weight of the protein sample, etc. So as we can see here, so these are the three dyes, wherein if you see their molecular weight, they are similar. So, and the major benefit of these dyes is they can be visualized in a different wavelength. Like if you see, like while like they can be emitted, like given its fluorescence ability by giving certain excitation and emission wavelength. So if you see, their excitation is at a different wavelength, because of which we can visualize all the three samples. So for example, you are having three samples, you have used one dye for one sample, one dye for another sample, and another dye for another sample. Then even after pooling them together, you can visualize them separately on the same gel itself because of their different excitation wavelength, and it will not affect the protein as such because it is pH insensitive as well, so it will not change the charge of the protein, etc. Because this dye doesn't get affected by pH. It is also photostable in nature, and it is spectrally distinct, indicating spectrally it has different excitation wavelengths, so it can be visualized differently. That's the reason cyanine dyes are used. Similarly, in terms of labeling, there are two different ways of labeling. Either, just a minute. Okay, I think I'll just change the camera. Okay, so in terms of labeling, we have two different types of labeling, the same dyes, but the technique you are using for labeling changes, wherein it can be either minimal labeling or saturation labeling. Generally, minimal labeling is used, but sometimes, depending upon the need, people also go for uh, saturation labeling too. So how exactly this dyes act in terms of labeling is they people use lysine for it. So people use in the sense, not like people use it. So dyes basically interact with lysine, and it labels around 3% of the proteins. And so as it is minimally labeled, so it is not affecting the protein structure, and uh, so it will not affect the migration also. Neither it is affected by pH. So whatever technique you are using, 2D, isoelectric focusing, and SGS based, those won't be affected because of the presence of these dyes. When the protein structure won't be affected. Similarly, it can interact with lysine, and lysine is a uh, hydrophilic protein, hydrophilic amino acid. So it's generally present on the surface of the protein, because of which the dyes can interact easily with the uh, dyes can interact easily with the lysine, and that's how labeling takes place. In terms of saturation labeling, cysteine labeling is done as the cysteine is present in greater amount, so dyes get labeled accordingly. Dyes is so dyes are used for labeling that specific protein accordingly. So, so lysine, generally minimal labeling is preferred, as I told you. So wherein lysine is used for lysine is preferred amino acid for labeling because its frequency is greater than 5%. So it is generally present in most of the proteins, because of which no matter what protein you are targeting and what protein you want to analyze, lysine is one of the best acid to choose. So while using DICE technique, as most of the protein will have lysine in it, it will be basically labeled with the help of lysine. And this lysine labeling, the way it interacts is it's at the end terminal alpha amino group, and it interacts with the lysine epsilon amino groups, and basically this interaction happens with the help of nucleophilic substitution. And in terms of minimal labeling, it only labels 3% of the whole proteome. So this is how the interaction takes place. So this amino group is from the lysine itself, from the whole protein, and that is something which interacts with the dye and which has a NHS ester reactive group. So if you see, it has a fluorophore. This is the fluorophore, and it has a linker. This specific part is the linker, and NHS reactive group, which actually interacts or reacts with the protein, and that's how labeling takes place. And it requires a specific condition for labeling. It requires pH 8.5 and at for 30 minutes at 0 degrees Celsius. That's how the labeling of the protein is done. And the amino group of lysine residue is positively charged at neutral or acidic pH. So at that's so the and dye is also positively charged. So the as these are neutral or positively charged and dye is also positive charged. So ultimately the net charge on Pi is effectively unchanged, and because of which it is not affecting the isoelectric focusing part, the first dimension. And both of these dyes, as you are using uh, for two different samples, they are spectrally resolvable dyes as well, and because they have similar mass to charge ratio or these both dyes, so it is not affecting the protein as such. In order to file like while preparation of samples while adding this specific dye, you basically dissolve these dyes into DMF initially, and this much amount of protein labeling, 50 microgram of protein labeling can be done with the help of this. So if you want to label this much amount of protein, you would require this much amount of dye, 400 picomoles of dye, which will be able to label around 50 microgram of protein. And if any reaction is, for example, if your protein is lesser in concentration, if you added more amount of dye, then you need to add free lysine to the solution itself, so that whatever dye is pending, which has not interacted with the protein, that will interact with the lysine, and you can remove the dye accordingly. So basically, you need to quench the excess amount of dye which you might have added while labeling the protein. So you need to add free lysine to it in order to remove that. So generally, we require internal standard while doing any kind of proteomic analysis. So what is internal standard? It is basically preparation by combining aliquots of equal amounts of all the samples basically in your experiment. So you need so while doing the sample preparation, extraction of the protein, everything you will be doing. So once you have the samples in hand, you need to take equal amount of those samples, like from all the samples. For example, your sample is around 1 ml, for example. So you need to take maybe 200 microliter from each and every sample and pool it together, and that pooled sample will act as your internal standard, or else it can be also called as QC, quality control, and that will help us in terms of analysis ahead. So whatever proteins are present in the pool, only those will be considered if any of the same proteins are observed in control or treated samples. So only those proteins are considered. For example, if any of the sample or control is giving some unknown protein which is not present in the pooled sample, you should not consider that specific protein, because that might be coming from any contamination, or it is not coming from your control or samples exactly, because pooled control will show you each and everything because they are already pooling all of these samples. So this is act as a standard, and you need to refer each and every protein band you find in this treated control sample with your internal standard, compare it with it, and if it is present, then only you can consider that protein. If not, you need to eliminate that. So that's the reason internal standard is required, and there are certain advantages of it. It reduces gel to gel variation. It helps in matching. That's what I was talking about, and comparison becomes easier. It acts as a reference point as well, and it while doing the analysis in the software, once you give the internal standard gel or the after visualizing the gel, you can automate accurate spot statistics with the help of that internal standard gel. That's how it would help. Yeah. So how do we prepare samples? So you have sample one, sample two, you are pulling them together, which acts as internal standard. So as we were discussing, there are three different dyes which can be used, Psi 3, Psi 5, and Psi 2. So Psi 3, for example, that is used for sample 1, Psi 2, Psi 5 is used for sample 2. You are pulling them together, making an internal standard before adding the dye, and that can be dyed with some other internal standard dye. If you have seen, these two dyes were having much more similar molecular weight as compared to that of Psi 2. So generally, Psi 2 is used for internal standard, and Psi 3 and Psi 5 is used for your samples. So once the you are mixing them with the dye, so the we have just gone through that protocol as well. This is how the protocol is for labeling. So once your labeling is done, you will mix them together and then load it onto the gel, perform isoelectric focusing, which we have already discussed. After performing isoelectric focusing, you will reduce it, you will alkylate it, and then perform SDS page, and you will get separated proteins. Now, as you added all three samples like pooled together and then added the sample. Now you can visualize sample one gel, sample two gel, as well as internal standard gel on the same gel itself. Like you can whatever proteins are present in sample one, whatever proteins are present in sample two, and similarly whichever like all of them will be present in your internal standard, but you can visualize them in this. Just a minute, people. Okay. So that was the first part of the lecture wherein Sir talked about how exactly digest perform. So now we studied in the last week wherein two-dimensional gel electrophoresis. After separating this, you need to stain the gel, right? Either using Comasi blue or silver stain or Procure diamond or other stains which we discussed. So in terms of dyes, you don't need any staining as such because your samples itself have been dyed already with the help of side dyes. So you can directly take this gel and visualize them by giving certain excitation and emission on those whichever is required for visualizing that specific sample. You can accordingly visualize them. So you can use, there are different scanners which we also discussed in terms of two-dimensional electrophoresis, such as Typhoon scanner, etc. So three images can be obtained from one gel itself, and you can analyze those images using Decider software. So there are different softwares available. One of the software is Decider software where image analysis can be performed. And there are different short forms or which are used in the software itself. So DIA indicates for minimizing gel to gel variation. BBA is minimizing biological variations. So this can be BBA can be like minimizing biological variations can be further analyzed using a tool called as EDA. So this specific tool uses different statistical tools like differential expression analysis, principal component analysis, pattern analysis, etc. All that can be done using EDA tool present in the Decider software. So this specific DICE technique can also be used for cell surface protein labeling. It can be used for labeling intact cells as well as labeling live cells, depending upon your experiment. So this technique can be utilized for both. And basically, those cells are incubated with side dyes, which we already studied in terms of the protocol. Then unreacted dye, you add lysine to it so that whatever is unreacted, that get reacted with it. And then you follow similar 2D and then uh, uh, do the analysis using Decider software. So if you are studying cellular proteins, you can label lyse cells as well. If you are studying cell surface protein, you can still label it and then use the similar protocol and perform this specific analysis. Generally, the gel images obtained are of this format, and as gel to gel variation will be minimized by using by visualizing all of these three images by different excitation emission wavelengths. So that was in the second part of the lecture, whereas in third part of the lecture, sir talked about analysis, how exactly dyes can be analyzed. So now you know like how exactly this has been prepared and they have performed 2D electrophoresis after labeling them, so it becomes dyes technique. And now you have visualized the gel by giving different excitation emission wavelengths. So when you are so Psi 2 is generally the standard as we mentioned. Psi 3 is red and Psi 5 is blue. So if you see visualize this, so in this you will see all of the proteins, whereas some proteins might be missing in the sample one and some might be missing in sample two. Then accordingly you can do an analysis. So for example, in this specific protein was getting overexpressed or getting expressed only in sample one but not getting expressed in sample two because that was treated sample, this was control sample or might be disease sample. So depending upon your experiment, it will you'll do accordingly such type of analysis and check which protein got upregulated or downregulated, etc. So this is just the overall procedure. Uh, you will collect the samples. Whereas if when we perform 2D gel electrophoresis, multiple gels you need to run for multiple samples. Whereas when you are doing a dice technique, you can pool them and accordingly perform. Like basically, you can take one control, one test, pool them together, have an internal standard, and run. Like so basically, number of uh, steps you need to perform will get reduced by using dice technique. So you can take control sample, test sample, everything you can pool it to one after giving certain specific dye treatment and then run one gel itself instead of running three different gels. Then using dyed imager decider software, you can then analyze them. Then while doing the analysis, you can see like if you can target that one specific protein bar. Like for example, if you want to see this specific protein, you can just select that and check whether it is present in the control, how much if it is present only in the treated ones, how much it is overexpressed, etc. So you can accordingly compare that. and do the analysis using the decider software. So both of these techniques can be planned depending upon the objectives you want to study. So although both of them are similar techniques, only thing is you can reduce gel to gel variability when you are using dyes, and similarly you can also what I so basically depending upon your objective, both of these technique can be used, and it is an extremely valuable tool that overcome gel to gel variations and cyanide dyes and can be run on a single gel itself, and you can check the differential expression in one gel, minimizing the load while doing the analysis. This is one of the case study. We already done multiple case studies in terms of two-dimensional gel electrophoresis. You can like for those case studies as well, dyes can also be performed. So according to your aim, when such type of analysis can be done. So while doing like sir talked about like while explaining diet applications, so this was the one example which sir talked about wherein so prostate cancer patients were taken and a diet analysis was performed. So in terms of this specific experiment or this paper which deals with prostate cancer, so they wanted to find a biomarker basically. Generally, uh, currently known biomarker is PSA, which is prostate specific antigen. This specific protein is uh, or generally present in higher concentration in the person suffering from prostate cancer, but it might also be present in healthy individual in some cases. So, so generally, this if prostate specific antigen is high in concentration, it doesn't necessarily indicate that a person might be suffering from prostate cancer. So this specific research paper or researchers were trying to find some other new biomarkers which can be considered as the main biomarker for detecting whether a person is suffering from prostate cancer or not. So they took healthy samples as well as person who was suffering from cancer. They took their serum samples, and we already studied that serum has albumin and globulin in our concentration. So they initially removed them, and those all are a part of sample preparation which we already done. Then they have, in terms of this study, they have divided into two sections, wherein the same disease sample, so there is no control sample in this specific gel or slide here. So they have used disease sample itself, but they have divided depending upon the stage wherein cohort 1 and cohort 2, their Gleason score is different. So what is Gleason score here? So it's basically stage at which that person might be suffering from prostate cancer. So depending upon their stage, early stage or later stage, a Gleason score is given, and so accordingly they had that metadata with them, and they have used that specific data and separated both disease patients. So they had cohort 1 and cohort 2, they mixed them together and done the differential gel electrophoresis analysis. And after doing the analysis, which we using decider software, they found that, for example, this protein got upregulated or might be only seen in one condition in one cohort but not seen in another cohort. Some of the proteins got upregulated, downregulated, etc. So they have used that analysis, and they found these two specific proteins, like is getting downregulated and this is getting upregulated, like Gleason 7 versus Gleason 5. So as compared to, so indicating that the person who had Gleason 7 in terms of prostate cancer, this specific protein was downregulated compared to the other one. Similarly, this protein was upregulated compared to the other one. So they found that these are the can be potentially biomarkers, but this specific analysis doesn't tell you about how much it is upregulated or how much it is downregulated. It just says that it is up it is up or down, but it will not tell you how much it is. So in order to validate that how much it is getting up regulated or down regulated, they have done ELISA technique wherein that specific protein's antigen was taken. So in the serum sample, this protein will be present, which is already detected here in proteomics analysis, and they found that that protein was getting down regulated in one condition compared to the other. So they took their serum sample, added onto the plate. You all might be knowing how ELISA works. Then there is a secondary antibody having certain enzymes. You add the substrate, and depending upon the concentration, that much amount will be visualized, right? So they found that Gleason 5 score had a much more up-regulated, like very high concentration of this specific protein compared to the other individuals here. So this is how they found that it is very high, and they whereas in terms of other protein, although it showed that it was upregulated in glycan 5, but it was not major. So there was not a major difference between them. So they figured out that this is the protein which can act as a potential biomarker compared to this protein. So they found that PEDF was a potential marker for early stage prostate cancer, and they confirmed differential expression of both of these proteins by way validation techniques, but they found that this is not much of a different, but this is like was way different compared to the seventh one. So that's how they found this can be a potential biomarker. So those were the one which were taken like explained in three parts of the lecture. I hope everything is good till now. Yes, SD. Should we move ahead? Yeah. So, so in week four, like in the week four itself, in third part of the lecture, we talked about system biology a bit. So, I already explained, but have you all have you heard about system biology anytime earlier? No. Oh, okay. So it's basically an integrated study wherein you are studying. So we are currently here talking about only proteomics as such, but when you want to do a complete system biology analysis, to do genomics, proteomics, transcriptomics. So because our body is very uh, like interdisciplinary in nature, if you see, like for a DNA to get converted into a protein, so that specific DNA replication also requires a protein like DNA polymerase. For converting into RNA, there are different transcriptional factors. There are different types of protein. There are different types of interactions being taking place. DNA with protein, protein with RNA, and protein with protein itself. So there are different types of interactions taking place, and studying each and every perspective is challenging. So generally, studying a system biology is challenging itself, and so so that was just an overview what system biology is all about. But uh, talking about giving an introduction of what proteomics is, and then we'll move ahead with uh, system biology. We already done a bit of introduction about proteomics, but in this lecture, part of the lectures are again revised a bit. So, so omics basically there are different types of omics. It can be genomics, transcriptomics, etc. Depending upon the molecule you are studying, like DNA, RNA, protein. Accordingly, such type of omics can be studied. It can be either global or cellular, indicating if you are studying only one type of cell and doing its transcriptomics or proteomics, it's called as cellular. Whereas if you are studying the whole system, all types of proteins, all types of RNA, or all metabolites, that's global. So how it came into picture? It's a set of proteins. If you're studying all of them, it's expressed by the genome. That's nothing a proteome. And if you're studying that proteome, it's called as proteomics, study of full set of proteins. And that proteins have different types of function. It is used for expression, localization, interaction, post-translation modification, etc. So studying each and every perspective of a protein is called that study is called as proteomics, basically. So before proteomics came into existence, there was genomics, and people initially tried to genome the whole sequence. So this is just a timeline how the genome sequencing were completely obtained for various strains. Whereas in terms of human genome project was completed around 2000 here, and it was started in the year 1996 approximately. So why proteomics? Because ultimately, whatever gene is being encoded, it is ultimately converted to the proteome, protein basically, and that's the reason studying proteomics is equally essential as compared to that of genomics and transcriptomics. Even those are completely essential because no, if we do not know the gene sequence, we do not know the protein as well. Similarly, how exactly gene is getting converted to RNA, because in RNA itself there are what do we say, introns being present, and there is a splicing which takes place in terms of trans a transcript. So ultimately, a functional protein needs to be coded out of DNA, which is done with the help of RNA at the middle stage after the transcription, and that is after splicing and stuff, translation is taken place, and then it is that specific code is functional in nature at a protein level. So each and every stage is equally important. But as currently we are studying proteomics here, so that's the ultimate functional part of that genetic code. And proteomics can be studied in different ways. We are currently studying only gel-based method, wherein we talked about 2D and DICE, wherein in both of this technique we talk about SGS page. We also talked about native page in week 2 or week 3 lectures, if I am not wrong. I think week 2 once, where we talked about blue native page as well. So as we were studying, like in terms of this technique, we require different dyes to be added, whereas in terms of dyes, you can use sign-in dyes directly to visualize the gel. So along with gel-based proteomics, there are gel-free methods as well using mass spectrometry. These are few gel-free based techniques. And mass spec, we'll be also talking about mass spec in week 5, like in the next week. So all those details about mass spec can be done in next week lectures. So along with that, interactomics and can be also be a part of proteomic study if you are trying to study how exactly one protein is interacting with another protein, or how one protein is interacting with some nucleotide or something, like all those come under a part of interactomics. And if you are just studying the structure of protein, the structural proteomics, generally one of the method which is used for studying the structure is exocrystallography, wherein your purified protein is crystallized and it is subjected to X-rays and it forms certain pattern, and that's how you can try and elucidate the structure of a protein, whatever pattern it makes after coming in contact with X-rays, and that's what helps in analyzing the structure of a protein. So, okay, so we were just doing that. Proteomics can be done by gel based, using mass spec, using protein microarray, label free techniques like SPR, etc. So all these techniques can be used for doing the proteomics analysis. So here, this is called as label free technique. You are not using any label. So it's the light which gets deflected depending upon the interaction taking place. So if there is no interaction, after interaction, it changes. So that's how you can determine whether your protein of interest is present or not. If it is present, how much it is present, etc. without using any label. So it is not affecting your protein as such, and I can still analyze your protein. So what system biology? So that's a general introduction of what proteomics is. So that's a very smaller part of system biology. System biology is a very huge concept. So it's basically examination of an biological entity itself, no matter human system, bacterial system, fungal system, whatever it is. But inside that specific system, as an integrated system rather than studying its individual characteristics. So you're not just studying protein or DNA or RNA or any metabolite, just that, but you're studying it in an integrated manner rather than just studying individual characters and its components. So it requires everything. The networks indicating interactomics, like signaling networks indicating one is giving signal to another, one that is giving signal to another neural system, for example, or cell-cell communication. All of this having there is a pattern of studying all of this. So if you are targeting one specific gene, for example, and you are studying each and every prospective of that specific gene, that's system biology, and studying system biology becomes difficult. So you need to have so you generally collect different literature from each and every part of it, each and every subsection, and try and combine it and make a conclusion out of it in order to perform the system biological study. So there are different levels of system biology. So it depends. Like you can uh, make a system within one cell itself, or you can make a system within the whole body. Like inside the cell itself, for example, you can make system like there is a nucleus, there is mitochondria, there is endoplasmic reticulum, there is Golgi apparatus, there is lysosome, etc. So inside that one cell itself, there are different systems, and you can study each and every system, how that is interacting with other system, how one is interacting with some other system. If you're studying that perspective, it's still a system biology. And that one cell is interacting with some other cell, it's called as uh, so this is within the cell, this is intercellular, indicating cell is getting communicated with some other molecular level, indicating DNA, RNA, protein, how they are interacting with each other, etc. And that whole thing in one specific system like respiratory system or nervous system. Or else if you want to study all these systems together, having some effect of this system on this system, or having some effect of this system into cardiovascular system, whether this basic cardiovascular system can be involved in all of these, right? So all of this is basically an interactive study rather than studying one component. So that's nothing but system biology. So why do we need system biology? Because that's how our any specific organism is, no matter if it is a bacteria or a human system, or whichever, and or if it is a living system, everything internal is interactive. So that's the reason we need system biology. So we cannot just study one component and have some conclusion that this component is responsible for this, etc. But how exactly that is responsible? We can only do that by studying transcript, by studying system biological, by having a system biological approach. Then only you can confidently make one specific conclusion out of one specific protein or gene sequence. So it's an holistic approach rather than reductionist approach, and it can be used for quantitative as well as qualitative biological data, and it generally makes biology a predictive science. Oh, I think uh, just a minute. Okay. So as we were discussing that, so as you need uh, different data initially, so it can be model analysis. So system biology can act in two different perspectives. You can, if you already have the data, for example, You create models out of that, use this data, and have like come up with certain biological conclusion out of that existing data which I was talking about. Like for example, that protein is genomics has been done, proteomics has been done related to that specific part. You integrate those two studies and try to find out like they came up with this conclusion, they came up with some other conclusion. You create model out of it and come come to certain conclusion using those different literature works. So that's this. But along with that, some people might think of, okay, this is happening, so this might be the reason. You do certain experiments, gain certain data out of that. So it is a circular system basically, and it goes on and on until we are and trying to understand the whole body itself. So it can be either model based or database. This is also explaining the same thing. Database indicating you trying to have creating certain hypothesis, do certain experiment, create data out of it. And model based, you have already having some prior information, some prior literature. You take up those all of those data, try and stimulate that data, create models, derive certain conclusion out of it. So, for example, if I give you in terms of model making, for example, uh, uh, if any organism is engineered for making some product, for example, no matter what product you are, for example, making some protein out of a system. So it got engineered, you use different promoters, you targeted some specific site, and you got certain expression. Now, accordingly, such type of studies might be there. Then you can try and make a model that if you use this specific promoter, if you use this specific site wherein it get integrated, and such type of like proteins have its own nature. For example, this type of nature proteins uh, are in like produce in higher yield compared to other type of proteins. So you give certain type of protein to the model itself, and it will predict that. Okay, if you are doing using such type of protein, then it might give you this much amount of yield or lesser amount of yield. So instead of that is what model based system biology is all about. I just gave one random example. So studying system components. So you can either study one protein, that is proteomics, either DNA, genomics, or interactomics, depends. And this approach can be studying system biology can have two different approaches, reductionist, or there is one more approach as well, which is called as the downward system, the bottom up approach, or up to below approach, wherein so in terms of reductionist approach, it is disintegrating the system into components and then studying them part by part. Like you disintegrate, you just study the genome, you just study the protein, you just study the RNA. Or if you talk about organelles, for example, you just study one specific organelle first and then the other organelle, how exactly they are interacting with each other. So part by part you will be studying. That's something called as reductionist approach. So this is called a systems information triangle, wherein uh, from molecular level to intracellular level to the whole organization. So it can be it's the same thing which we seen molecule, intracellular, intercellular, and the whole system or the whole organism. So these are the molecular level. So those molecules get interacted within the cell itself. Those are acts as metabolic pathway regulatory motifs. And when they communicate with other cells, intercellular, that's functional modules, and that's how the whole large scale organization happens. Like the whole system is functional. So this is one can be one of the approach for studying system biology, wherein extraction and mining of complex and quantitative biological data. So you already have the existing data, you extract that, try to have some conclusion out of it, integrating them together and then analyzing the data by using creating models, by having certain mathematical equations to it, or just developing a mechanistic approach out of the literature available. And then validating them as well. Like whatever models you created out of that, you validate that using the existing literature. So this cycle goes on and on. You have a database or model based, and it's this cycle gets in circle itself. This is what system biology triangle is. So different approaches to generate data. You have the data, then try and understand the system with the help of the data by part by part. So each and every section is important in order to complete the whole system study. Like generally, researchers are in this, like we as researchers, we are in this section where we are doing different genetic modification, creating certain hypotheses, trying to prove that hypothesis, doing certain experiments. The people who have done bioinformatics, they are in this part of the system biology where they take up the existing data, try and create models out of it, make certain conclusions using those data. And there are system sciences as well, which work on interacting between both of these together, wherein synthesis, analysis, and modeling concept is done. The system study requires data set from different approaches, genomics, proteomics, and transcriptomics, etc. And when we have that data, we can try and do certain experimental validation or model validation, and then give a conclusion to a system in a broader perspective, which is nothing but system biology. So it requires integration or collaboration of, so studying, as we were discussing, that it's challenging and it is not a job of one specific individual. So one might study only gene, one might study only protein, one might study the interaction between gene and protein. So by doing all of this, then we can study the whole system together. So we require biologists, we require statisticians, we require physicists, we require chemistry people, engineers, etc. So all of them, we require a collaborative effect in order to understand one specific system. So this is what system biology, it requires everything in order to understand the whole physiology of an individual. So that's the reason it is extremely challenging understanding the dynamics of biological networks by modeling, simulation, etc. And it requires some statistical, mathematical inclusion into understanding the whole system. So that's the summary of part 4 of this lecture. Then in part 5, he went ahead by explaining more about system biology, how exactly that can be used. We'll talk about that in like 5 minutes. We can take a break of 5 minutes and then talk about that. Okay. In terms of what? Precision medicine. Yeah. So, so it depends. For example, you are talking about precision medicine, indicating that uh, if a person is suffering from any specific. So, are you talking about personalized medicine, which is uh, okay, 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 got it. Okay, yeah. So, like if yeah, exactly, exactly. Yeah, so basically, if you so the amount of because this specific system is very complicated, and we never know what is that one specific thing which might be affecting. So considering each and every perspective will make your much model much more stronger. Like you need to add one parameter uh, to your model and try and validate it by adding each and every parameter one by one, and then that's how you can make your model much more stronger, basically. Okay, okay, great. Exactly. Okay, so basically, I am not into modeling. I am into experimental biology, basically. So I know that something like modeling exists, but I am not an expert related to that. How exactly those specific things are performed, but I have a like broad overview idea like how exactly uh, modeling works, etc. Like you are into the like that's what system biology is. We need both of us. Like I as an experimentalist, you need data from me, but using that data, predicting something, you can do that much better than me. Like yes, exactly. Yeah, that's what. Okay, okay. Okay, so don't get confused by the word. Basically, it is the study is been given a specific name like system biology. What you are doing is nothing but a part of system biology itself. What I am doing is nothing but a part of system biology itself. So ultimately, you will be using that specific part. So doing a system biology indicating that uh, you have data of everything, like each and every component of the whole system, and integrating that whole system together, you can have one system biological conclusion related to it. But generally, people work in parts of it. So as I said, like don't get confused by that specific word system biology. You don't have to consider that whole organism as system. You can consider that one cell itself is also a system in itself. So that's the reason even smaller studies use the word system biology. If you have seen certain papers, they like there might be a system biology word itself might be written in the title itself. So they are studying that one specific system, like for example, Krebs cycle itself, for example, glycolysis itself. That's a system. And you're trying to study each and every part of that system. That can also be a part of system biology. Although, in a broader perspective, it's just a smaller part of the whole system. But that specific smaller system itself, people still call them a part of system biology. And people like the wordings which are used in the paper, etc., they can still consider that as system biology. If I give you an example of like for us in terms of experimentalist, uh, so for example, if have you heard about Krebs cycle anytime? Yeah, yeah, TCA cycle. So for example, it has one of the component, for example, succinate. If I say so, succinate requires. So for example, for overexpressing succinate, you are over like for in order to get succinate as your product, then succinyl coenzyme A, etc. All those you will try and express in a greater amount, like over express those specific enzymes, so that or else block the further one. For I think after succinate there is fumarate. So whatever is the enzyme which is involved for converting succinate to fumarate, yeah, yeah. So then you can basically block the expression of that specific protein or knock out that specific enzyme and overexpress the earlier one so that you get your product succinate in higher amount. So I'm just giving an example. So yeah, so that's what I'm saying, trying to tell you. Like even that specific smaller part of the system can still be considered as a system basically. Like that cycle is a system, and you're targeting that system. And people, if I'm trying to publish a paper out of this, so I'll still call it as system biological engineering of that strain and trying to get succinate out of the uh, that specific organism, etc. I can still call this as a system biological study. As I'm studying Krebs cycle, I'm trying to block one thing, I'm trying to enhance one protein, I'm trying, or else if you want to uh, like express it extracellularly, then you need to bring that succinate out of the cell. Then you'll have certain transporters, adding transporters to the organism, and then it will come out, etc. So you're still studying a system like in a smaller approach, but that's still a system. Don't get confused by the word itself. You can apply this word in an entire way or else in a smaller way as well. Yeah, so that's how it is. I'll just drink water and come, and we'll start in like 2, 3 minutes for the next part of the lecture. Yeah. Okay. So in terms of last part of the lecture, sir digged in more into system biology, wherein yeah, you still wanted to ask something. Okay. Yeah, yeah, yeah. So like for example, for RX, so what the person who is doing just studying genomics sequencing, still it is a part of system biology. Just studying 2D page, it's still a part of system biology. Like in terms of just the proteomics, these are different proteomics technique. This is the interactomics technique. This is kind of used for metabolomics a bit, whereas these are used for transcriptomics technique, etc. These are the one which are used for genomics technique. So these are all technologies employed for studying system biology at different levels of the whole system. Gene level, genomic, transcriptome level, transcriptomics, proteome level, proteomics, metabolomics, there is phenomics as well. So in terms of building models, it is a circular loop basically. You have like the system itself doing certain experimental analysis, creating a model out of it. Like this is where we work, like in the real system. perform experiments, etc. We try and get the data, and bioinformatics people will try and use that data, create certain models out of that using the data, do certain predictive sciences using this, and this is a loop which goes on. It's a different way of explaining the same thing which we were discussing in the initial slides. So they have it like there can be different levels in terms of building models. Just for example, level one is just gene expressional study. Level two, like that's inside the cell. Maybe level two is between the interactomic study, intracellularly, like within the cell itself. Level three is intercellularly, studying between the cells, how exactly that interaction might be taking place, how information is carried out from one to another. Level 4, studying the whole system together. So it has different levels by in order to, by in terms of building models for system biology. So this is a bioinformatics approach wherein you take the data, you align them, like for example for gene sequences, align them together, have some predictive science, create certain prediction out of the existing data, etc. Similarly, for this is in terms of gene sequence. Similarly, it can be done for the protein sequence itself, and try and develop models out of this. So this is the modeling scheme. So this is a cycle which goes on, like in uh, experimentalists, we engineer the strain, change the gene of the uh, mutate the gene, and try and check whether if it is having certain effect or not having an effect. And all of these changes can be measured by different techniques: spectroscopic technique, imaging technique, arrays, whatever proteomics measurement, like dyes, everything, like all different types of measurement techniques. Then that's how our data will be produced out of doing certain experiments. By measuring them, that's how it will create a data. Then that specific data can be used for mining, which is a part of bioinformatics. Then that specific data which was created, which will be helpful for creating models. It depends upon what type of model you want to create. It can be of reaction models, it can be mechanistic models, statistical models, stochastic models, etc., depending upon your aim. So uh, all these are basically an integrated part of the one specific system, which is system biology. So no matter if you are working in this category, still you are a system biologist. If you are working in this category, still it is a part of system biology. No matter if people are working in this category, all this are a part of system biology together. So in order to uh, compile all of them and then coming out to conclusion, that's also a part of system biology itself. If you don't have this, then you won't be able to do the entire conclusion out of one specific system. So again, the same thing but explained in a different way. Having biological system performing experiment, having the data, or you have created a model by having the literature which is already being present, initial parameters which you know, you stimulate that and get the stimulated data indicating predictive data, because you have built certain models using existing literature. Then you predicted that, stimulated it by giving certain condition to it, and check whether it is producing something of similar content or not. Then you have that stimulated data as well. You can do a comparative study, which was actually observed in the experiment and what you got by the stimulation. That's how you can validate your model as well. And this system goes on and on. So it's like this. Then two different types of models, like ordinary differential equations. So this is if you are including mathematical equations to your modeling, and that might help in making your models much more accurate by including statistics and mathematics to it. Stoichiometric indicating network based study, interactomics is involved in this. Then there was a small discussion by one of the professor from Indianapolis, I think so, wherein he tried to discuss about uh, like the system biological study itself, majorly dealing with network, majorly focused on interactomics, which is a part of system biology, and he discussed that part of the system biological section. So initially talked about, just gave a brief introduction of central dogma, how DNA is converted to RNA, where the transcription factors, etc., are involved for successfully completing converting it into RNA. Then similarly, ribosomes, how those ribosomes are involved in making the whole protein, how these transcription factors can actually regulate the expression of one specific protein if it is binding to an inhibitor or a promoter, etc. That interaction of binding between the protein with the gene, etc., with RNA, and that will actually regulate the expression of the protein. So all those part of interactomics basically. So in order to study these networks, uh, this is how they are like shown basically. So each and every part or a molecule is called as node, no matter protein, a metabolite, or a transcription factor, whatever, that's the node. And if it is having some interaction with the another protein, if it is giving some information to another or helping in converting it to something else, those are predicted, like depicted by links. So if this protein is predicted by, like, given a link with another, so they have some properties together. Either they are interacting, either they are sending some information, etc. So this is how the network in biology is like shown in terms of pictorial representation. So if you see such kind of a image anywhere, it is showing that one molecule is interacting with another molecule. It is also interacting with some other. It is also interacting with some other. So that's how this node network is shown. So it's also called as a graph, basically, which is a collection of nodes and links. So basically, nodes and links are generally used by physicists or computational biologists. So we basically core biologist people have to borrowed these specific terms from uh, them and try to like use those terms in terms of understanding the interactomics part of biology. So these graphs can be either undirected or directed. Generally, for protein-protein interaction, they are undirected, and whereas in terms of translational or uh, interaction, like metabolite interaction, those are directed ones, because directed indicating one is influencing the other is influencing the other. So the if that way of interaction is called as directed interaction, and if only two are involved in it, like they are interacting, that's undirected one. It is not influencing that specific interaction to happen something else. So that's undirected interaction, whereas directed one is, this is influencing this so that it gets interacted with the other one. So it has a direction basically. If this is not getting interacted with this, then this will not interact with this further. So it has some direction to it that's directed. For example, we were talking about Krebs cycle itself. So one product will be formed if that specific enzyme is involved. It is given certain direction, right? So that's directed basically. Uh, there are different parameters as well, like degree, path length, and clustering to this representation of interaction. So, wherein you can see there are three things involved which are influencing one of the protein, for example, and that can interact with multiple proteins. Path length indicating how exactly this is influencing the another one. Clustering coefficient. So it depends, and we can find this clustering coefficient depending upon this neighbors of node and links, between the neighbors. So this is the formula basically. So if you see here, this specific fluorescent protein is interacting, like green one, has three nodes which it like the neighbor nodes, but they are not linked with each other. They are just interacting. So M is 0 here and N is 3. So if you apply this to this specific formula, the clustering coefficient comes up to 0. Whereas in terms of if this is how the interaction is taking place, if you see there are two of this interaction. If you see here, these blue ones, so that's the link between the neighbors of node, which is by m is equals to 2. And if you apply that to the formula, the clustering coefficient is 0.66. That's how, like depending upon the type of interaction, you can talk about clustering coefficient. It tells about how interconnected are the neighbors of a given node. So if you see here, there is no interconnection happening between the neighbor nodes. That's the reason this clustering coefficient is 0. Whereas here, it is completely interacting, like each and everything is getting cross reacted with each and everything. So that's the reason it is completed. It's 1. So it tells us about the interconnected neighbors of a given node. Then this is about transcriptional network, how exactly transcription gets influenced and is responsible for interaction. So this specific study can, like, if you know how exactly one is getting interacted with another, how is the whole cycle present for one specific molecule. That specific can be study is used for drug targeting as well. So if you see in terms of this network, this middle one is the most essential one, and that is something which is interacting with each and everything. If you see this, if this is interacting with this gets interacted with this, etc. So if you are targeting that specific uh, part, it might affect the whole cycle itself. So generally, with the help of this network study, you can also try and plan how what can be your specific drug target so that drug will be effective in nature. So that's how this node link study can be helpful in order to determine the drug targets. This is similarly talking about transcription factors which actually activate or influence the or regulate the expression of a protein, and that's how can accordingly help you to design drug targets. Then a structure of transcriptional regulation. So there are different ways of regulating or different motives. So this is a basic unit. Generally, if you see, there is one transcription factor which will target the gene and influence to whether to express or block the expression of the gene. But there are different motives to it in terms of local level. Uh, that is, for example, one is influencing. Uh, so here, if you see, one is influencing the other protein as well as two other protein. Whereas here there are two transcriptional factors uh, influencing two different factors altogether. Whereas here this is kind of singular one, whereas one itself can influence different types of proteins altogether. So this there are different ways, patterns of interconnections or patterns of motives present in a specific system. And if you know about, uh, for example, for this specific transcription factor itself, and if you know about how the transcriptional factor works, that will help you in terms of understanding that interactomics exactly, and accordingly it can be used for different applications. So here just talked about one of the application of drug targets. It can be used for upregulation of a gene or downregulation of a gene or blocking the gene, etc. If you know how the interaction is taking place inside the body. Gene regulation is a highly regulated and complex process, which we all know, and that's the reason system biological study is important. So one person can study one part and another can integrate all of them and uh, say that this is how it is happening inside the body, and we can understand the system much more effectively. Then Sir talked a bit of RNA binding protein, which is essential for transporting. So this yellow one is RNA binding protein, which is essential in transferring the RNA from the nucleus to the cytoplasm, so that now it can be uh, used for removing the introns and converting that into the protein. So, so I talked a bit about this specific RNA binding protein and how change in that specific protein can lead to different diseases as such. So here uh, it was shown like these are orange ones are the diseases, and these are the if I think these are the transcriptional factors, the blue ones, and that is influencing the proteins expression regulation. So if these are affected, ultimately a protein is getting affected, their expression, etc., and that can lead to different disorders, and it is interconnected altogether, which is a network of RNA binding proteins. And if this system is not working properly, it can lead to diseases accordingly. So, so in terms of drug discovery, for example, in an healthy person, this is how the interaction takes place. So, for example, this is one protein interacting with another protein, this is interacting with another protein, and that's how the whole interaction is. And whereas in terms of this is just an example, random example basically, and how drug targets can be designed if you know this interactomics study properly, and if you have that network uh, in front of you, then accordingly choosing a drug target becomes easier. People generally believe on magic bullet stuff, like you just randomly targeting one thing and hoping that it will interact similar to that of healthy state. But that generally happens in a very less possibility of happening that. So having a network pharmacological study helps you all to decide which specific protein can be targeted and convert that disease state into uh, healthy state again. So this is how the cellular interactomes, like they might interact with each and everything. It depends upon protein-protein interaction or protein metabolite interaction or a protein nucleotide interaction. So you can need to study each and every type of interaction, how exactly that specific part is getting influenced inside the system itself. And if you are targeting that specific part of the interactomics, perturbation indicating changes which are happening because of the presence of drug, then you can ultimately come with some conclusion out of it that if you target that specific protein, the interaction will accordingly change and it will come to healthy state or it might affect more, etc. All of those conclusions can be we can come up with that. So this study can be done for drug networking, target networking, disease networking, etc. So overall, this was majorly about uh, networks and model makings in the week, like part 4 and part 5 lectures, wherein network based approaches are essential for dissecting and designing the biological system. So this will only happen when you so this is broader way of studying system biology. What I was doing and what you are doing in terms of that, those are uh, not a broader level of doing system biology. So this network-based approaches are generally can be considered as a broader way of understanding system altogether, because you are studying the interaction between each and every part, how exactly that protein might be binding with another protein and influencing the expression of something else altogether. So they play an important role in biomarker identification as well, and you can target a specific drug out of that. a specific target for one specific drug and having some uh, influence of like working of that specific drug, whether it is effective or not. So all those were discussed in these 5 lectures. So should we go ahead with question and answers? Yeah. Okay, so let's do it. So you need to analyze these two graphs basically. So this is specific, basically a protein A, and this is how, while doing the analysis, they found that with Psi3 it was something like this, and with Psi5, in Psi5 sample, it is looking like this. So if you see, Psi3 dye was used to identify the control sample, and Psi5 was for tumor sample. Below is the representation of that specific protein, for example, this specific protein. So you need to analyze which ones are correct in terms of while doing the analysis. So expression of protein A is unchanged. So it can be clearly seen that it is changed here. So first option is obviously wrong. So it is either protein A is upregulated in tumor sample or protein A is downregulated in tumor sample. You can analyze that clearly which one is getting upregulated. And protein A is differentially expressed in control and tumor sample. So even this is obviously true, it is differentially expressed, it is not same. So we need to decide whether 2 is right or 3 is right, and 4 is obviously right here. So you can guess which one is the correct option. So I'll stop sharing and reshare so that I have that window wherein you can write it in the chat box and I can see that. Yeah. So which one is the correct option? So this is a tumor sample basically, and this is upregulated. So it's 2 and 4 and not 3 and 4. Just minimize this. Okay. So now this is the gel images, like a collection of samples with two distinct conditions, time point A and time point B, respectively. He intended to use Geiger experiment, label those samples with time point A with sci-fi. So time point A was done with sci-fi and those with time point B was done with Psi3. The internal standard was Psi2. Then SGS page analysis were collected for from both pools of individuals labeled samples. So they mixed them together. Then the gel was examined to identify the protein bands afterwards. This is a combined image basically. So consider one lane as one image, second lane as another image, third lane as another image, fourth as another image, fifth as another image. It's just that excitation and emission wavelength was different and accordingly those were visualized here. The combined image of gel was produced after scanning to for necessary wavelength is depicted below. So now you need to identify different how gel 1 to 5 should be matched with various runs of run labeled samples, various types of run labeled samples. Yes, so uh, I'll just give you a hint. If you see here, uh, this has everything. The fourth lane, you can answer like any of this A, B, C, D. So basically, if you see in this fourth, you can see everything like all the bands together, indicating this is an internal standard which has all of them together, which might be with Psi2 dye, the internal standard. The fourth one would be probably Psi2, right? Because it has all the bands. So this is a pooled one, pooled sample, so it should have everything, whatever might be present in point A and point B together. So if you see for a fourth one, which is side 2, so if you see, we have option number 4, option number 3, and option number 2. So we can eliminate the first option here. And then if you have gone like we have gone through the lecture wherein sci-fi and sci-3 it is uh, red and green in color. So this is reddish and green. So sci-fi will be red, wherein if you see, so this we have already eliminated. So the first one is sci-fi. You can mute yourself, like it is creating certain disturbance. Whenever you want to speak, you can unmute and speak. So if you see here, this is sci-fi, this is sci-3. So it is like in terms of this option, sci-fi and sci-3. Whereas when you mix all of them, like when you visualize all together, it is whitish. So if you see the third option showing all three together, and when you visualize only the samples and not the internal standard together, it is showing in yellowish color when red and green mixed together. So it's sci-fi and sci-3 together. So it's the fourth option which is correct. That's how you can analyze this gel and give answer. Similarly, which of the following does give images yellow and white bands? So we just discussed that how do we get yellow and white bands? So, but we can find the answer accordingly here. So proteins that are present in both sample from time point A and B are represented by the two colored bands here. So because they are present in both samples from time point A and B are represented in two colored bands. Do you think this is right? This is generally talking about the internal standard first thing because they are they are present in both the samples. Proteins that are present in time point A sample are represented by both colored bands, or only the protein present in the sample of time point B are represented by yellow bands, while the proteins present in the sample point time point A are represented by white bands. But that's not the case because psi-5 and psi-3, this is time point A and time point B. Similarly, it's the opposite, only the protein present in sample time point A are represented by yellow, whereas B are represented by white. So these two are obviously not correct according to me. You need to decide on either first or second. Which one do you think? So it's present in both the samples. So it's present in both the samples, time point A and B, which are represented by two colored bands. So as psi-5 and psi-3 both are together, so it is giving the color accordingly, both mixed together. So that's the reason it is white and yellow. For a tumor sample, so for the tumor sample, Sci-Fi was used. For control sample, uh, Sci-3 was used, and she also created an internal standard with Sci-2. So internal standard indicating mixing this tumor sample and control sample together, pooling them and labeling them with Sci-2. Then merging the sample, all of them were merged together. SDS gel was performed and collectively tagged with these were run. The gel was scanned after it at three different wavelengths of Psi2, Psi3, and Psi5, and this is how the image was obtained. So, which of the following contains only control samples? Control samples uh, indicating this control sample Psi3. So, this is internal standard basically, which has everything 1 and 6. Psi2 is blue in color. So, that's what pooled one is to pool sample. So, we need to find Psi3. Psi5 is reddish. So, this is having red. So now we need to find the question is only control. So you need to see the lane in which there is only green. So this has mixture of colors. This has mixture of colors. I think this even this ninth one has red also. So even that is mixed together. So I can see only 8 and 2, which is having only control sample. So if we look at the options, it's fourth option wherein this gel is only showing green bands indicating only control sample is present in 2 and 8. Similarly, we need to predict which dye has been depicted in lane 5 and 10 of the gels, which are utilized for labeling the samples. So lane 5 and 10, can you answer lane 5 and 10 here? So if you look at lane 5, it has bluish, it has pinkish as well, like red and white mixed together, and whereas here, like we can clearly see that this might be having all three together, whereas this is having two samples together. So if we try and analyze this, so lane 5 has all the three, whereas lane 2 has two of them. Lane 10, side 2, side 3, side 5 together. That's the reason it is showing all the colors, whereas this was visualized using two excitation emission wavelengths. So that's what the answer is. So what is the degree and out degree of red and yellow nodes? So I think I have not explained this. So for example, if you are selecting this, this is the in degree, and this, for example, for yellow, this is one in degree and three are the out degrees. So that is influencing other three. You need to see the direction here. So this is the in degree and this is the out degree. Similarly for red, so you need to find how much in degree and out degree are present for red and yellow. So is it 1, 0 in degree and out degree? For I think it's first is red and then it's yellow node. Okay, can you answer this question? What? 1, 0 and 1, 0. How come? Like, can you explain that? So for red, it is 1, 0, yes, because it is in degree is 1. There is no out degree here. There's nothing going out. For yellow, it is 1 and 3, right? Yeah, 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 yeah. So similarly, there are three networks A, B, and C listed below. Find the red dots individuals clustering coefficient. So we just discussed that clustering coefficient formula. So you need to go through that formula and try and find the coefficients and accordingly. But still, even if you don't have that formula, by looking at the interaction, you can still answer the question. Just look at the red ones, okay, and check how they are. Find the red nodes, individual clustering coefficient for each network and arrange the network on the basis of clustering coefficient. C would be greatest, right? So it's the highest. Yeah, it will give you one, right? Because it is completely interacting. Like we need to calculate the coefficient for these two. So you need to calculate basically. So yeah, so it's showing that A and C are same. So like I'm not sure how exactly it is, but maybe you need to find the coefficient for this, like having N and M values, and we can come up with the uh, answer. So maybe even this is giving you one as an answer because of which A and C are together. So apply that formula and uh, you will be able to get this answer accordingly. So three different types of networks are typically investigated in system biology. Which of the following, when applied to three these networks using graph theory, is correct for general classification? I told you that one is undirected, whereas other two are directed, and we. So which are the other two which are directed, whereas one thing which is undirected? Yes. So three gel pictures are given here. Which method from the list below would have been used to obtain these gel images like this? Third, three 2D gels stained with fast green FCF, Nile red stain, and Comasi stain. Is that so? It's this, right? Because you will be pooling the samples together. Then when you visualize them, only that specific sample will be visualized. I think there's some problem with this gel here. It should show each and everything because Sci2 is generally bluish in color and it is a pooled sample together. So basically this gel should show each and every band present in image 1 and image 2. I think there's some problem with this gel. That's the reason you might have answered third as an option might be. But generally giving these three colors is generally uh, Dye's experiment. side 2, sci-fi, and side 3, sci-fi, and side 2. So side 2 should generally give you all the bands, which is an pooled control by comparing image 1 and image 2. So here, because of that specific gel image here, you might have answered this, I feel so, but the answer is Dij. So given below are few steps involved during gel-based proteomics experiment. How will you do? Which of the following is best describe the gel-based technique? What is the first step? What is the second step? Or like which all steps are involved in one-dimensional electrophoresis, two-dimensional electrophoresis, and Dij? So for 1D, like for one-dimensional, what all steps we use? So these are the, I think I need to zoom out a bit more, but I cannot zoom out more. So I can show you just three options. Which one? B option. So let's check it once and then I can go for the answer. So one dimension we use 4 and 7, SDS and gel staining, yes. For two dimensional 3, 4, 5, and 7, two dimensional isolatory focusing 4, 5, and 7. Okay. For dyes use 1 to 7 or 1 to 6, they say. So answer is 4, 7. So it's C option, 3, 4, 5, 7, and 1 to 6. As I mentioned, like we don't require gel staining here because you already uh, 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 stained or like dyed your samples, right? By using Sci2, Sci5, and Sci6, you can directly visualize them using because they are fluorescently tagged. You can take the gel, give certain excitation emission wavelength. You don't have to do gel staining as such. So it's 1 to 6 only in terms of dyes, and you don't require to do gel staining. Yes. Yeah, so that was it in terms of week 4 question and answers. So, still, if you have any doubts, you can ask me. I think we are already up with the time. It's 8:59. Yes, do you have any questions? Okay. So, yeah, okay. Thank you for attending. So, mm, we can meet next week with, so next week we'll be talking about mass spectrometry. Uh, like it's gel-free based technique. So it will quantify your protein as well and tell what exactly the protein is by using mass spec, whereas in 2D or digest, you just know the molecular weight, that's it. Like this might be a protein, but in order to validate that and prove it that that is your protein, you need mass spec. So that's the benefit of mass spec. So we'll be talking about mass spec in the next lecture. Okay, thank you for attending. Yeah, yeah, thanks.